Welcome to the deep dive. So glad you could join us.
Good to be here.
We've got this really interesting source you sent over, a paper from, wow, way back in 2001, New England Journal of Medicine.
Yeah, classic.
And our job, as always, is to dig in and pull out the key insights, the really important stuff, just for you. So today, we're looking at this pivotal clinical trial. It was for a new drug targeting a specific blood cancer, chronic myeloid leukemia, CML.
That's right. And pivotal is the word. I mean, our goal here isn't just to summarize. We want to understand what they found, why it was such a big deal then, back in 2001.
Right.
And, what it really hinted at for the future of cancer treatment. A whole new approach.
Okay, I'm ready. This sounds like a story about finding a cancer's Achilles heel.
Pretty much, yeah.
Okay, let's set the scene first. Before this new drug, STI-571, what was the deal with CML? What is it, according to this paper, and what were doctors working with back then?
So CML, the paper describes it as a bone marrow thing. Basically, your myeloid cells, a type of white blood cell, they just start multiplying like crazy, uncontrolled expansion.
Okay.
And it progresses, right? Starts chronic, can go on for years, but then it often shifts gear to an accelerated phase, and then blast crisis. That's the really aggressive end stage, where the cells just don't mature properly.
And treatments. What did patients have?
Well, the options weren't great, honestly. The source mentions interferon alpha. It could, stretch out that chronic phase, but the side effects were rough. Really rough. I think constant flu, fatigue, depression. A lot of people just couldn't tolerate it long term.
Okay, so interferon. What else?
The only shot at a cure was an allogeneic bone marrow transplant.
Risky, though.
Hugely risky. You're wiping out their whole immune system, replacing it, high chance of complications, even death.
Yeah.
And that's if you could even find a matched donor, which, wasn't always possible.
The tough choices. Limited effectiveness or high risk?
Exactly. A real bind for patients.
Okay, but then there was this discovery, something about the genetics. The Philadelphia chromosome. That sounds important.
Oh, absolutely critical. This is like the linchpin for the whole story. Scientists found this specific genetic abnormality, the Philadelphia chromosome, or Phi chromosome, in almost all CML cancer cells.
What is it? Like a broken chromosome?
It's a swap, actually, a translocation. A piece of chromosome 9 breaks off and swaps places with a piece of chromosome 22.
And that swap does something bad.
It creates a monster, basically, fusion gene. And that gene makes a fusion protein called BCR-ABL. BCR-ABL.
Okay, BCR-ABL. Why is that the problem?
Because BCR-ABL is a type of enzyme called a tyrosine kinase. Think of kinases as signal relays in the cell, telling it when to grow, divide, survive.
Like switches.
Exactly. But the BCR-ABL kinase is what they call constitutively activated. It's stuck in the on position permanently. Yeah. It's constantly yelling, grow, divide, don't die, at the cell, ignoring all the normal stop signals. And the research showed this one protein, BCR-ABL, was enough to cause CML, and its kinase activity, that on switch, was essential.
So if that one faulty enzyme is the main culprit.
Right. The big question became, could you design a drug to just switch off that specific enzyme? Could you target the root cause without, nuking the whole system?
Enter STI-571, later known as Gleevec or Imatinib. This was the drug designed for that job.
Precisely. They found STI-571 through screening lots of compounds, looking specifically for something that would block kinases. And its mechanism is pretty neat.
How does it work?
It's a specific inhibitor of that BCR-ABL tyrosine kinase. It works by basically plugging up the spot where the enzyme gets its fuel. ATP is the fuel.
Okay.
STI-571 fits into that ATP binding site, like jamming the wrong key in the ignition. ATP can't get in, so the enzyme can't rev up and send those rogue signals. It shuts it down.
And it was specific, didn't hit too many other things.
That was the beauty of it, according to the early work. Highly specific for BCR-ABL, and just a couple of other very similar kinases, like PDGF receptor and KIT, much more targeted than traditional chemo.
And the lab tests before this human trial, they looked promising.
Very promising. The preclinical data showed STI-571 could make CML cells stop growing, or even trigger their self-destruct program apoptosis.
But it left normal cells alone.
Largely, yes. Because normal cells don't rely on that faulty BCR-ABL signal. So the idea was, hit the cancer where it's different, spare the healthy tissue, a huge potential advantage.
Okay, so the stage is set. They take this promising drug into a phase 1 trial. What was the setup? Who were the patients?
Right, phase 1. So primary goals, safety, figuring out the right dose range, how the body handles the drug. They enrolled 83 patients, all with chronic phase CML, all confirmed FIP positive.
And the key thing.
The key thing. These patients had already been treated with interferon alpha, and it had failed them. Meaning either they were resistant, their CML came back, or they just couldn't handle the side effects anymore. So these folks were really running out of options.
Got it. And the trial design, dosing.
Dose escalation. They started patients low, 25 milligrams a day, orally, like a pill. Then in different groups, they carefully increased the dose cohort by cohort, all the way up to 1,000 milligrams daily.
Trying to find that ceiling, the maximum tolerated dose.
Exactly. See how high they could go safely. Yeah. And patients stayed on the drug unless they had bad side effects or the CML got worse. No other cancer drugs allowed during the study.
And how are they tracking success or problems?
Standard stuff, mostly. Lots of blood tests, checking blood counts, bone marrow biopsies to look at the cells directly, and cytogenetics, counting the percentage of cells still carrying that Philadelphia chromosome.
That seems like a really direct measure of whether it's working on the root cause.
It is a deeper response than just seeing blood counts normalize. And they also had that clever way to check if the drug was hitting its molecular target in the patients.
Ah, yes, the CRKL protein. Remind me how that works.
Yeah, CRKL. So BCR-ABL, when it's active, sticks a phosphate group onto CRKL. It phosphorylates it.
Okay.
So they took blood samples, used a lab technique gel electrophoresis to separate the phosphorylated CRKL from the unphosphorylated kind. If the drug, SCI-571, was working and shutting down BCR-ABL,
then you'd see less of the phosphorylated CRKL.
Exactly. You'd see the phosphate tag disappear, showing the drug was inhibiting the enzyme right there in the patient's cells.
Neat. Okay, before the big results, how did the drug behave in the body? Pharmacokinetics. And did that CRKL test confirm target engagement?
Yeah, the pharmacokinetics looked good. The drug was absorbed well orally. They measured blood levels, found it reached steady levels with daily dosing, and importantly,
yes,
the trough levels, the lowest point between doses for patients on 400 milligrams or more, were consistently higher than the concentration they knew from lab studies was needed to inhibit BCR-ABL by 50% and kill CML cells.
So the drug levels in people looked high enough to actually work.
Correct. It suggested they were hitting therapeutically relevant concentrations.
And the CRKL assay, did it back that up?
It did. Beautifully. At the really low doses, not much change in CRKL phosphorylation. But once they got to 85 milligram and up, they saw a clear decrease in phosphorylated CRKL and an increase in the unphosphorylated form.
The drug was switching off the target.
Yes. And that effect seemed to max out, sort of plateau, in the 250 to 750 milligram dose range. It was strong evidence that the drug was doing biochemically what it was designed to do inside the patients.
Okay, all right. The moment of truth then. Safety first. How did patients handle STI-571, especially considering interferon was so tough?
This was one of those striking findings, really. The drug was generally very well tolerated. So well tolerated, in fact, that they didn't actually identify a maximum tolerated dose, or MTD, in this study.
They couldn't push the dose high enough to find a limit? That's unusual for chemo trials.
Very unusual. It suggests a wide therapeutic window.
So what were the side effects then?
The most common things reported were relatively mild, usually grade 1 or 2. Things like nausea, some muscle aches, bit of swelling or edema, and diarrhea.
Manageable stuff, mostly.
Yeah, for the most part. There were some more significant things like drops in blood counts, anemia, low platelets, low neutrophils, especially at the higher doses. And some patients had elevations in liver enzymes.
But were these showstoppers? Did people have to quit the trial?
Very few did, considering only 2 patients who were getting 300 milligrams or more had to stop the drug permanently because of side effects. And incredibly importantly, the paper states there were no deaths during the study attributed to STI-571.
Wow. Compared to the risks of transplant or the misery of interferon, that safety profile alone is remarkable.
Absolutely. Game-changing level of tolerability.
Okay, now, the big one, efficacy. Did it work? Did it fight the leukemia in these patients who had failed interferon? Let's start with the blood counts, the hematologic response.
This is where jaws really dropped back in 2001. The results were, frankly, stunning. For patients getting 140 milligrams or more per day, 100% had a hematologic response.
Every single one?
Every single one. And if you look at the group getting 300 milligrams or more daily, that was 54 patients. 53 of them, 98%, achieved a complete hematologic response.
Meaning their blood counts went back to normal?
Completely normal. White counts, platelets normalized, spleen sizes reduced. Just a normalization of the blood picture.
And quickly.
Yeah, typically within just 4 weeks. Remember, these are patients whose CML was progressing despite interferon. Now, within a month of starting this pill, 98% have normal blood counts.
That's incredible. What about that one patient who didn't respond initially at 350 milligram? There was a drug interaction story there.
Ah, yes, good point. That patient was also taking phenytoin, an anti-seizure medication. Phenytoin is known to rev up certain liver enzymes, including one called CYP3A4, which metabolizes STI-571.
So is clearing the drug out too fast?
Exactly. Lower drug levels. Once they figured that out, stopped the phenytoin, and bumped up the STI-571 dose slightly, that patient also achieved a complete hematologic response.
Wow. Shows how important those drug levels are.
Definitely. And it highlights real-world complexities.
Were these responses lasting? Did the blood count stay normal?
For most, yes. The paper reports that of the 53 who got a complete hematologic response, 51 maintained it for the duration of the follow-up, which was a median of about 265 days, nearly 9 months. Only 2 lost their response during that time.
Okay, impressive hematologic control. But what about that deeper level? The cytogenetics, reducing the actual FET-positive cancer cells.
Right, the cytogenetic response. This is hitting the disease at its genetic root. And here, too, the results were significant, though not quite as universal as the blood count normalization.
What were the numbers?
Okay, again, focusing on patients getting 300 milligrams or more daily, 54% of them had some level of cytogenetic response, major or minor.
And major response. That's a big reduction.
Yes, a major response meant the percentage of FIL-positive cells in their bone marrow dropped below 35%. And 31% of the patients in that dose group achieved a major response.
Okay, nearly a third. And complete. Any parents have the phenylketonuria chromosome disappear entirely?
Oh, yes. This was perhaps the most profound finding. 13%, 7 out of those 54 patients, achieved a complete cytogenetic remission. No detectable Philadelphia chromosome-positive cells in their bone marrow analysis.
So in over 1 in 10 patients, the defining genetic marker of the cancer was gone.
Essentially, yes. Eradicated, at least to the level of detection they were using. And they even mentioned doing more sensitive molecular tests like PhilH and PCR for the BCR-ABL message on a few of these complete responders, and found they were negative even by those highly sensitive methods, suggesting a really deep level of response.
Yes, wow. Okay, let's step back. Phase 1 trial. Supposed to be mainly about safety, but these efficacy results, especially in patients who had failed prior therapy, this had to be huge news.
Oh, it was earth-shattering in the oncology world. This study provided incredibly strong evidence that STI-571 wasn't just safe, it was dramatically active against CML. It validated the entire concept of targeting BCR-ABL.
Yeah.
And, that observation about how the blood counts normalize gradually, not like a rapid kill-off from chemo, was interesting too. It made them wonder if the drug was maybe restoring some normal control mechanisms, allowing healthy cells to outcompete the leukemia rather than just being purely cytotoxic.
A different way of working.
Potentially, yeah. And that might explain why the deeper cytogenetic responses took a bit longer to emerge.
And what are the dose for future studies, since they didn't find a toxicity limit?
Right. They didn't have an MTD to guide them, so looked at other factors, the pharmacokinetics showing good drug levels were achieved, the CRKL data showing maximal target inhibition around 750 milligrams, and the fact that hematologic responses plateaued at higher doses.
And putting that together,
they reasoned that a dose of at least 400 milligrams daily seemed optimal to recommend for future, larger trials, aiming for that sufficient drug level and target inhibition.
Makes sense. So the big picture takeaway. This wasn't just about a better drug for CML, was it?
Not at all.
Okay.
I mean, yes, it was transformative for CML patients, but the broader significance was enormous. This study was arguably the most compelling proof of concept up to that point for the entire strategy of targeted cancer therapy.
Find the molecular flaw, design a drug to hit it specifically.
Exactly. It showed you could potentially get profound anti-cancer activity with far less toxicity by understanding the specific biology of the cancer cell. It really ushered in the modern era of personalized oncology.
Incredible. What a journey through just one paper. We've seen how zeroing in on that single molecular defect, BCR-ABL, and hitting it with STI-571 brought about, well, unprecedented results for CML patients who had run out of road.
Absolutely. This 2001 study wasn't just a step forward. It was like a leap into a new paradigm. Targeted therapy, personalized medicine. This was the poster child that showed it could really work, and work dramatically well.
So, for you listening, what's the lasting message here? This study is over 2 decades old now, but its impact
is still felt every day in oncology. It laid down a blueprint.
And maybe leaves us with a thought to chew on.
Yeah, I think so. Consider this. CML, driven largely by this single, powerful BCR-ABL engine, turned out to be exquisitely sensitive to a drug targeting just that engine. The success was stunning. But most other cancers aren't quite like that, are they? They often have multiple genetic drivers, they evolve, they develop resistance.
Much more complex targets.
Right. So the provocative thought is, given the huge success in this relative simpler case, what does it tell us about the true scale of the challenge and maybe the potential rewards of applying this targeted approach across the incredibly diverse and complex landscape of all human cancers? How do we translate this principle when the targets are multiple, moving, and intertwined?
That's definitely something to mull over, the promise and the complexity that followed this breakthrough.
Indeed.
Thank you for joining us for this deep dive. We'll catch you next time.